All right, so today we are going to go over meiosis. Now, meiosis is going to be pretty much the same process as mitosis with a few differences. And you guys can see here, I drew the cell cycle for meiosis, and we still have prophase, we still have metaphase, we still have anaphase, and we still have telophase, but we have them twice, okay? So let's go through this, and then I'll go through some differences between the cells produced, and then we'll go over all the individual phases. So, just like mitosis, we are still going to start with the cell growing from it being produced the last time. After that, we're still going to replicate our DNA. We're still going to get that chromosome number up to 92, just like we did with mitosis. Remember, we start off with 46 chromosomes, and we're going to get to 92 after the DNA is replicated or copied. After that, we're going to make sure that DNA has no mutations in it, okay? Now, here's the difference between meiosis and mitosis. In meiosis, our goal here is to produce sex cells. In other words, for males, produce sperm. For females, produce egg, okay? So what we're going to do here is we are going to go through 2 cell divisions instead of just 1. So we're going to go through prophase, metaphase, anaphase, telophase, all with the number 1 after them. And then we're going to go into prophase, metaphase, anaphase, telophase, all with number 2 after them for a second division. Why we're doing this is when you are being made, when you start off as a single solitary cell, that's you, okay, in the womb is 1 cell. That cell has 46 chromosomes, okay? In mitosis, the cells that we produced each had 46 chromosomes at the end. However, here's the difference. Whenever we are making you, the sperm comes and it meets the egg, and they combine together through fertilization, and they make you with 46 chromosomes. Each one of these is going to give you 23 chromosomes. So the egg is going to have 23 chromosomes, the sperm is going to have 23 chromosomes, and those combine together to make you as one single solitary cell with 46 chromosomes. At the end of mitosis, the cell had 46 chromosomes. We want the end cells of meiosis, the sperm and the egg, to have half that number so that you have a correct number of chromosomes when you are 1 cell, okay? So that's why we're going to go through 2 cell divisions. When we are doing meiosis, we always have to put a number after prophase, metaphase, anaphase, and telophase. All right, so we know that we are referring to prophase 1 or prophase 2. If you don't see a number after it, if it just says prophase, it's referring to mitosis, okay? If there is a number after it, it's referring to meiosis. All right, so let me go over some differences between the cells produced here. So for mitosis, and if you remember I went over this in the last video, we have 46 chromosomes in each cell. In other words, we could say the cells are diploid. They have 2 of each chromosome, okay? In meiosis, we are only going to have 23 chromosomes in each of our cells. And these are going to be haploid. So I did a small rendition of a karyotype last time in the mitosis video. Let me show you the difference on what they look like. So if we're talking about chromosome number 1, we talked about you getting one from your dad and your mom. And in chromosome 2, same thing, you get one from your dad. It'll be a little bit smaller because as we get to the bigger numbers, the chromosomes decrease in size a little bit. You get one from your mom, one from your dad, okay? In meiosis, here's what it's going to look like. You are going to choose, now you don't actually do this, but you give one of your chromosomes to that cell. So number 1, we're only going to have 1 chromosome. Number 2, we're only going to have 1 chromosome instead of how they looked like in mitosis. This is considered diploid because we have 2 of each of our chromosomes. This is considered haploid because we only have 1 of each of our chromosomes. All right. Okay. The other thing, mitosis is going to deal with our body cells. Meiosis is going to deal with our sex cells. This is how we make our sex cells. Next, we are only going to go through one division for mitosis. Meiosis, we're going to go through two divisions. Okay? And those are your main differences right there between the cells produced for mitosis and meiosis. The big one is you got to know that they are diploid and haploid. Another thing I want to mention, sometimes they'll abbreviate diploid as 2N, meaning they have two copies of each of the chromosomes, chromosomes being the N, the variable. Haploid would be N or 1N. Sometimes you'll see it abbreviated like that, okay? All right, so what I'm going to do now, guys, is I'm going to go through prophase 1, metaphase 1, anaphase 1, telophase 1, all right? We'll go through all those different steps, and I'll show you the processes just like I did in mitosis on the last video, all right? So let me erase all this. Sorry for the dead air. And I'm also going to go over the differences between meiosis and mitosis whenever I'm starting to draw all the individual phases out. Okay, so let's start off with meiosis number 1. Meiosis number 1 is going to consist of prophase 1, metaphase 1, anaphase 1, and telophase 1. I'm just going to write this Pro 1, Met 1, Ana 1, and Telo 1. Okay, so same thing happens in meiosis 1 for prophase 1 as happens in prophase of mitosis. The nucleus disappears. Nothing changes there. We're going to have those centrioles. They're going to start to move to opposite poles. Nothing changes there. Here's where our differences are going to occur. So, let me do a small karyotype here again, just like I did last time. We'll have one from mom and one from dad. Let me do number 2 as well. Prophase number 1, guys, this is going to be our longest one out of all of them. We'll say purple is from mom, and the brown will be from dad. Okay, so the goal of meiosis is to allow you to make a kid. So when we are doing this, if these chromosomes were given to you, they're actually from your mom and dad. You're going to pass one of those chromosomes onto your kid. So when you think about it, if you were to give this chromosome, so these are yours, if you were to give this one to your kid, that kid's going to look like your mom or their grandmother, okay? If you give this one, they're going to probably look more like your grandfather, all right, or your dad. So here's what we're going to do to make sure that the kid has some genetic variation. In other words, so the kid looks a little bit different than your grandma or your grandpa. So there is a huge step in prophase 1, and this is called crossing over. Here's what happens. So these chromosomes are already duplicated, but we're also going to have, so if you remember they're duplicated like that, we're also going to have pieces of this one break off. And let me replicate this one. We're going to have pieces of this one also break off. And what's going to happen is they are going to interchange these traits between one another. So that way, your kids are getting genes from both of your parents, not just one of them, in that one chromosome that you are giving to that sex cell. All right. And there's going to be more than just one place. It's going to be a bunch of places here. We'll have some red over there, some blue over there. In other words, guys, these are just going to be completely swapped everywhere in different places. So that way we have a nice blending of our two homologous chromosomes. And the same thing is going to happen over here with our purple and our brown. We're going to have pieces get cut out there. Put some brown over there. Put some purple here. Okay. The goal of crossing over, guys, is to increase our genetic variation in the next generation. Okay? Now, I'm not going to draw all this. I'm not going to keep going, and swapping my chromosomes around, and it would just take a very long time. So, I'm still going to draw them as X's, just like we did in mitosis, but know that crossing over occurs. Know that crossing over occurs during prophase 1. Okay? Now, I'm going to go on to metaphase 2 here, and there's only one difference. I'm sorry, metaphase 1 here. There's only one difference between metaphase 1 and regular metaphase of mitosis. And here's that difference. So we're going to have the centrioles. They're here. We're good. They're going to connect to our chromosomes. Here's our difference. If you remember, I drew them as straight X's going all the way down. So for a metaphase of mitosis, it looks like this. We got our X there. We got our X there. Purple one there. And there's our blue. I have way too many markers in my hands, but okay. So that's how it looked like in metaphase. Here's what it's going to look like in metaphase number 2. So it's going to be metaphase number 1, sorry. It's going to look a little bit different. So instead of them lining up as those X's, they're actually going to line up with the homologous chromosome. So here's what it looks like. Okay. So they are literally on top of one another. What we call that is a tetrad. Tetra means 4. Okay. So the tetrads line up in the middle. There's your only difference. That's it. Next one, anaphase. We're going to start to reel it in just like we did in mitosis. But instead of separating into the chromatids, we're going to stay as our X's here. There's that one there. That one there. Boom. And boom. Okay. Now, it's going to be completely random which one goes to which side. We could have had the blue one go to that side. We could have had the red one go to that side. Okay? It happens. It's completely random. And what that does also, guys, is that increases our genetic variation again by not the same one going to each side every single time you make a sex chromosome. All right, telophase. So we are going to now, at the end, I'm just going to draw them as 2 cells, pretend like cytokinesis already happened. We're going to have our nucleus reform here. And let's put our X's in each one. We're going to have the red one there, brown one on that side, blue one here, and our purple one right there. All right, cool. So now, let's get into Meiosis Part 2. So here's going to be prophase 2. Metaphase 2, Anaphase 2, and I'll just keep them under the one so you guys know what they're doing here. And then we're going to have Telophase 2. Now, what I'm going to do is I'm only going to take one of these cells and bring it down. I'm not going to bring both of these cells down, okay? Actually, since my purple's kind of running out, I'm going to bring that one down. But just so you know, the red one, red and purple chromosome one, is also going to go through this same process of meiosis number 2, okay? So just so you know, while this one is going through what we're going to go through, the other one is doing the exact same thing, all right? Okay, so nothing crazy here, guys. We are not going to have crossing over happen again. We're going to have our nucleus disappearing once again, and we are going to then have our blue one in there, our brown one in there, and our centrioles are going to move outward. Pretty much it. Metaphase number 2 is actually going to look identical to metaphase in mitosis. The only difference is we're not going to have as many chromosomes lined up in the middle. So we're going to have our blue one, our brown one. That's it for metaphase 2. Okay, so as you can see, they line up in the middle just like in metaphase of mitosis. Next one, let's reel them in. We're going to separate them into chromatids. And then our last step, we are going to have 1, 2 cells at the end. Okay, so 2 cells, we'd have 2 cells from here, so we would get 4 cells overall at the end of meiosis number 2. Okay, so that's all your steps of meiosis. One thing I want to mention, guys, is when we are making these might be sperm, these might be egg. When we are making sperm, it's called spermatogenesis. Every time we go through this, we make 4 sperm cells. Now, the opposite of that is oogenesis, okay? Some people call it O, some people call it oo, whatever. But at the end of making the egg, we are actually going to have 1 large egg cell and 3 smaller cells. We call them polar bodies, right? So sperm, we're going to get 4 at the end. We're still going to get 4 for the female version to make the egg, but the 1 egg is going to be huge, and then the other 3 cells are not going to be big. They're not going to be real eggs. Like I said, they're called polar bodies. All right, so this is the end of meiosis, guys. This is the end of us making our sex cells, and this is also the end of cell reproduction. Next time, we're going to start to get into genetics, and then we'll finish up with evolution. All right, have a good day, guys.